Okay, so as we were mentioning in class, anatomy is really broken up into several types of anatomy. You have kind of down to the cellular anatomy or microanatomy, and then you have histology, which is the study of tissues, and we'll talk about that in the next lecture. And then there's the gross anatomy, which is what we'll spend most of our time this semester talking about. But to start, I wanted to just go through some cellular anatomy, and this will be helpful for you, especially when you get to physiology and when we start talking about some of the organelles and the plasma membrane and things like that within the cell. So this will be a foundation for physiology next semester as we talk about what goes on inside the cell. So if you think about the cell, it's the smallest structural unit in the body that performs all vital functions. And there's two main types of cells in the human body. We have sex cells, which we also call germ cells. And of course, you know them as maybe sperm and eggs. The proper anatomical term would be oocytes for the female germ cells. And then anything that's not a sex cell or germ cell, we would call a somatic cell. And you're going to see this word somatic probably multiple times during the course. And it will be, sorry, just trying to fix this pencil. It will be a word that refers to the body. So it comes from the Greek word soma, which means body, human body. So we'll see this word somatic over and over, and it'll be kind of contrasted. Here it's contrasted with germ cells or sex cells. Later on we'll see it's contrasted with visceral or organ cells. So the term somatic really means body. So somatic cells and sex cells or germ cells. Within the cell, we have two basic components. We have the plasma membrane, which separates the cell from any external environment. So in the body, that's going to be separating the cell from the interstitial fluid that the cell floats in. All cells have to be surrounded by fluid in order to survive, but you want to prevent that fluid from getting into the cell anytime it wants to, and that plasma membrane kind of forms a separation. And then you have what's inside the cell, and that can be separated into the fluid that's inside the cell, which we call the cytoplasm. The cytoplasm is actually the fluid plus dissolved ions and proteins and other material that's dissolved in the liquid. The cytosol would be just the liquid part. Looking at what may be in the cytoplasm, we're going to look at some organelles. Now, we're going to talk about in this class the idea of organs and organ systems. So, for example, the cardiovascular system, and the main organ is the heart, or the respiratory system, and the main organ is the lungs. Here we're talking about organelles. Organelles are kind of like little small organs that perform a specific function, and we're going to look at several of these organelles and what function they perform inside the cell. Organelles themselves can be broken up into organelles that have a membrane, kind of like the cell here has a membrane, and organelles that do not have a membrane. So we'll call them membranous and non-membranous organelles. So we'll go through and look at several examples from each of these two categories, membranous and non-membranous. But let's get back to the plasma membrane. You're going to see in physiology that the plasma membrane is super important in kind of separating what goes on inside the cell from what goes on around the cell. It also is really important in directing the flow of material both into the cell from the external environment as well as from the cell out into the blood supply, for example. And this plasma membrane is going to be really important when you get to physiology. So let's take a quick look at it. So it does provide physical isolation, which I've been talking about. And as I mentioned, it does regulate the exchange, particularly of ions, water, perhaps hormones or other proteins that are being made inside the cell. The plasma membrane really directs the traffic of things into and out of the cell. There are we're going to look at the plasma membrane and what composes it, but what we won't talk a lot about in this class, and we'll talk more about in physiology, is that embedded in the plasma membrane are proteins that act as receptors, either receptors for hormones or neurotransmitters, or they may be carrier proteins that allow hormones or other proteins to enter into the cell and change the function of the cell. So we would call that intercellular communication really occurs at that plasma membrane oftentimes as well. And then the plasma membrane, obviously, since it's the outside of the cell, would be involved in holding two cells together. So if I want two skin cells, for example, to be attached to each other, that attachment is going to occur through the plasma membrane, and we'll talk about what some of those attachments look like. It will also bind each cell to the underlying tissue. So for example, we're going to talk about epithelial tissue, let's say the epithelial tissue of your skin, and that has to be connected to the deeper tissues of the skin so that your skin doesn't slide off your body. And of course, those connections are also made through the plasma membrane. Okay, so primarily when we talk about the plasma membrane, we use this term. We call it a phospholipid, as you can see there, bilayer. And we call it a bilayer because there are two layers of phospholipids, and this becomes really important. And again, when we get to physiology, you'll see much more why it's important. But for this course, I just want to make you aware of the kind of the construction of this plasma membrane. So if we just go in a little bit closer, when we talk about phospholipid bilayer, we're talking about these guys here with their tails. So the circles are the phosphate part, and the tails are the lipids, so phospholipid. And you can see that it's a bilayer because you have phospholipids here on the outside, and you also have phospholipids here on the inside. And they form kind of a sandwich with the phosphates on the outside and the lipids here on the inside. Swimming among the lipids, you'll also find things like cholesterol. That's these yellow components here. And cholesterol just kind of helps the plasma membrane remain a little bit more fluid, helps it not to get too stiff, gives it extra flexibility. But it's this phospholipid bilayer that really keeps most material that is floating around out here in the extracellular fluid. It kind of prevents it from coming into the cell because most molecules and proteins and these things are either too big or they're afraid of the fat. We call that lipophobic, where they really don't like interacting with the lipids in the membrane. Now you can see some other things in this plasma membrane. You can see that most of it is formed by those phospholipids, which are kind of in a sandwich, that bilayer. But you can see there's also some proteins embedded. So we've got proteins like these, which are either channels for ions or proteins to come in and out, or maybe they're transport proteins. And again, we'll get into more of that in physiology, but just for now, understand that there's a phospholipid bilayer. Within that bilayer, you may find things like cholesterol, sterols, you see here. There are some proteins embedded in that wall. Some of those proteins are helping transport material into and out of the cell. Some of those proteins are helping to connect one cell to another. Some of them are acting as receptors for hormones and neurotransmitters. So there are proteins embedded throughout the plasma membrane. And there are also some glycolipids or glycoproteins, and you can see them kind of sticking out. They function to kind of signal to immune cells that this is a normal body cell so that they don't get attacked, for example, by the immune system, and they have some other functions as well. Okay, membrane permeability, and this is going to be more important again in physiology, but just to give you a foundation here in anatomy, membrane permeability means what is allowed to enter or leave the cell, and that is controlled by the plasma membrane. So some substances are able to pass through without a specialized transport protein, for example. It's not a lot of material, but some material. So, for example, very small things like gases, so oxygen and carbon dioxide, for example, can just diffuse through that plasma membrane without much problem. So, things that are small, things that are lipophilic, meaning that they like to mix with fats, they will be able to dissolve into that phospholipid bilayer and enter the cell as well. But there are many substances, and probably most substances, that are not allowed to pass through the membrane. So we say that the plasma membrane is selectively permeable. That means it's not permeable to everything. It's only permeable to some things, and it has special transporters inside of it that help it to become permeable to material that couldn't naturally cross the membrane. So here are some factors that affect the permeability of the cell membrane. I've already mentioned the size of the substance, so things that are very small, for example, will pass easily across that membrane. Electrical charge, a lot of the proteins on the surface of the cell are negative and will tend to repel other negatively charged material. Molecular shape, and so, for example, those proteins that we saw embedded in the phospholipid membrane, they are uniquely shaped to receive or accept only particular proteins that can fit inside their shape. So, depending on the shape of the molecule, it may or may not fit one of those carrier proteins, in which case it would not be able to cross the membrane if it doesn't fit one of those carrier proteins. And then a big one is going to be lipid solubility. So those materials that are lipophilic, philic meaning liking, right? Philadelphia is the city of brotherly love. So philic comes from the Greek word philos, which means to love. So if it loves fat, if it mixes well with fat, then it's going to have a better chance of entering the cell through the plasma membrane. If, however, the material is lipophobic, and you know phobic like a phobia, a fear, so if it fears lipids or does not interact well with lipids, then it's going to have a hard time entering the plasma membrane. So what kind of causes this selective permeability? Well, we've already been talking about this, but the composition of the membrane is huge. So because the membrane has those phosphates on the outside, but that sandwich layer of lipid in the middle, things that do not interact well with lipids will have a hard time passing, as I mentioned. So those that are small and lipid soluble, we already mentioned, cross the membrane freely. And then there are certain transport molecules. We saw on that picture previously of the plasma membrane, we saw those blue proteins embedded in the plasma membrane. Those are carrier proteins or channels, and they will allow certain molecules to pass into the cell through them. There's two types of transport that occurs with these proteins that are embedded in the plasma membrane. There are those that transport material actively, meaning it requires energy. And there are those that do it passively, which means there is no energy required. So passive transport is going to be something that occurs without energy, and active transport will require energy. Let's look at some examples of passive transport and then active transport. So passive transport, again, requires no energy. We would call this diffusion. So, for example, gases I had mentioned earlier, oxygen and carbon dioxide, they will flow freely across the plasma membrane. They don't require any particular transport mechanism. They will diffuse into or out of the cell simply based on the concentration gradient. So in the picture that you see here, you've got a lot of carbon dioxide, these gray circles, inside the cell, and relatively few carbon dioxide molecules outside the cell. So diffusion always occurs, always has movement of molecules from high concentration to low concentration. So in this particular example, the carbon dioxide is highly concentrated inside the cell and has a low concentration outside the cell. And because it's small, it will be able to diffuse freely across the plasma membrane, and it will go from inside to outside, so from high to low. And that requires no energy. That's excuse me, that's the natural movement of those molecules by diffusion. Another form of passive transport is really just a modified type of diffusion called osmosis. So osmosis is the diffusion of water. So diffusion can refer to any substance that moves from high to low concentration. When we're talking about water in particular moving across cell membranes or across tissue compartments, we're going to use the term osmosis. And so water will move from low solute concentration to high solute concentration. So it's a little bit of an opposite way of thinking about diffusion. Normally we think of diffusion from high concentration to low concentration. When we look at water, it's still true that the water is moving from high concentration to low concentration. So if we look at the picture here, inside the cell there is more water molecules. If we just took a ratio of water to solute, the green is the solute. So there are way more water molecules than there are solute molecules. So we might say that the concentration of water inside the cell is high, and the concentration of water outside the cell is relatively lower. So all diffusion moves from high concentration to low concentration. So the water will move to outside the cell. And that is a fine way of thinking about it. You could say osmosis is the movement of water from a high concentration of water to a low concentration of water. However, practically speaking, when we look at the movement of water, typically we're looking at the solute concentration. We're saying how high is the solute concentration inside the cell and outside the cell. So again, in this example, if you're looking at the green circles, that's the solute. Let's say it's an ion like sodium. So we've got a lot of sodium outside the cell. You can see all the green circles, and relatively low sodium inside the cell. And so water is going to see that high solute concentration outside of the cell, and it's going to want to create an equilibrium so that the concentration inside the cell equals the concentration outside the cell. So the way that it can change the concentration is by dilution. So if you add water to a solute concentration, it will cause that concentration to become more dilute or less concentrated, right? So if outside the cell is more concentrated and inside the cell is less concentrated, water is going to move outside the cell in order to create a concentration that's equal inside the cell and outside the cell. So in this case, we could say that the water moves from low solute concentration here inside the cell to high solute concentration outside the cell. So for osmosis, water tends to move towards high solute concentration. Okay, and the last kind of passive transport I wanted to mention is when you have to use some kind of a carrier protein, like those ones that we saw embedded in the plasma membrane, like you can see here in the picture. So sometimes in passive transport, material will move freely across the plasma membrane. Sometimes the molecule is too big, for example, or too lipophobic to naturally cross the barrier. In that case, it's going to need some kind of a carrier protein. So let's look at glucose, for example, here in this picture. Glucose is a relatively large molecule compared to something like oxygen or carbon dioxide. So it's too big to freely diffuse across the plasma membrane. So there are carrier proteins in many cells that help to transport glucose from the interstitial fluid into the cell so that your cell can use the glucose to make energy. But it's still passive transport because there's no energy required for this movement. Because the glucose concentration is always, or nearly always, higher on the outside of the cell, that means that by the laws of diffusion, remember, diffusion wants material to move from high concentration to low concentration. So if the glucose is high outside the cell, by diffusion, it's going to want to move inside the cell where the concentration is low. Now, it's too large to do that across the membrane, but if it happens to bump into one of these carrier molecules, it will be able to be transported across the cell membrane and into the cytoplasm. And it requires no energy because the concentration gradient here on the outside is greater. So the energy comes from that difference in concentration gradient. And so no energy needs to be added in order to transport the glucose across the cell membrane. It does require a carrier, but it does not require any extra energy being added. Therefore, it's still passive transport. You might say that when there is a high concentration outside the cell and a low concentration inside the cell, there's a type of potential energy that's built up because that high concentration on the outside wants to flow across the cell and enter the cell in order to create an equilibrium. However, it's prevented from entering the cell by that plasma membrane, unless, as in the case of glucose here, it finds a carrier protein that it's able to attach to and then make that trip across the plasma membrane. And because there's all that potential energy built up, no energy is required to make those molecules move. They naturally move towards low concentration. So it's still passive transport. Let's look at some active transport examples. Active transport is often used to transport ions into or out of the cell. So we'll learn this more in physiology, but as an example, in order for electric signals to work in the body, we need to have a high concentration of sodium on the outside of the cell, a low concentration of sodium inside the cell, and conversely, we need to have a high concentration of potassium inside the cell and a low concentration of potassium outside the cell. So how do cells maintain this concentration difference? Well, they do it through pumps. And pumps means it's going to require energy. And because it requires energy, it's going to be a form of active transport. So in order to maintain that concentration difference inside and out of the cell is going to use pumps to pump sodium out of the cell and to pump potassium inside the cell. And you can see here in the picture, we call this the sodium-potassium exchange pump, or just the sodium-potassium pump. Sometimes we call it sodium-potassium ATPase because it uses ATP in order to pump ions against its concentration gradient. So, for example, out here in the interstitial fluid, there's already a high concentration of sodium. So if you want to move sodium from inside the cell to outside the cell, well, that's unnatural. The sodium doesn't want to go in that direction. In fact, the sodium would like to come into the cell and create an equilibrium, an equal number of sodium ions inside and outside the cell. So if you're going to move ions against its concentration gradient or opposite to its concentration gradient, you're going to have to put in some energy to make that happen. And that's where this ATP comes in. And this is why we call it a pump. A pump takes energy. And then the same thing is true with potassium. So you're going to pump out sodium and you're going to pump in potassium, and it's going to require energy in the form of ATP. So we're going to say this is active transport. And this sodium-potassium pump you're going to see is very important when you get to physiology. Other forms of active transport are things like endocytosis. So the term endo means inside, right? And the term exo means outside. And cytosis just means cell. So if we have endocytosis, that means we want to bring things inside the cell. If we have exocytosis, that means we are expelling things from the cell to the outside of the cell. So endocytosis has two primary forms. We have pinocytosis, which is basically cell drinking. So that's just the cell taking water in through a vesicle. And then we have what's called phagocytosis. Let's see if we can find the word here. There it is up there, phagocytosis, which is going to be cell eating, where you're going to take not only fluid, but you're going to take in some substance. Maybe it's a bacteria, maybe it's a protein that you're going to actually envelop into a vesicle and carry that into the cell. So pinocytosis is drinking, just taking in fluid. Phagocytosis is taking in some kind of a substance in addition to the fluid. And often we're talking about something like a bacteria. These also come from the Greek words pino means to drink and phago means to eat. So that's where these words come from. So the next one is just showing exocytosis, which is moving things out of the cell. So very similar process to endocytosis, but it's working in reverse. This might be, for example, hormones that are made inside the cell that need to get out of the cell. That might happen through exocytosis. Other proteins that are made inside the cell that need to be released into the blood supply, that might also happen through exocytosis. So microvilli are another form of non-membranous organelles. They're formed from these microtubules as part of the cytoskeleton of the cell. So you can think of the inside of the cell as having a skeleton, much like our body has a skeleton. And that skeleton inside the cell gives the cell shape. It also provides means for transport, kind of like a railroad system for things to be moved along in different directions. Microvilli, as you can see in the picture, are kind of extensions of the plasma membrane. So you see the membrane here, and then it kind of comes out in a finger-like projection. And you can see that that's being formed by these filaments that are kind of pushing that plasma membrane out. What's really important to understand about microvilli is their function or their purpose, and their purpose is to increase surface area for absorption. That's really the biggest takeaway from microvilli. So they're going to be found in parts of the body where it's important to be absorbing material from the surrounding environment. So I think the small intestines is a good example because you have nutrients flowing through the lumen of the small intestine, and you need to absorb those nutrients into the blood supply. And so the more surface area you have available for absorption, the more of those nutrients can be absorbed from the small intestine. So that's one of the places that you'll find microvilli. The kidneys is the same. There are a lot of things that need to be absorbed, reabsorbed, secreted in the renal tubules, and so you'll find microvilli in there as well. Excuse me. The cytoplasm we mentioned briefly before. Cytoplasm is basically everything that's inside the cell. And we can break that apart into the cytosol, which is the liquid part and its dissolved particles, as well as the organelles. And so I kind of mentioned this on the picture before. But what I want to make clear here is that the cytosol, and I mentioned this briefly previously, is that the cytosol inside cells is high in potassium. This is going to be really important in physiology when we talk about action potentials and resting membrane potential. So for now, just keep in your mind that the cytoplasm is high in potassium. The extracellular fluid, meaning the fluid surrounding cells on the outside of cells, is actually going to be low in potassium and high in sodium. And that's equally as important. So this difference in the concentration of potassium on the inside and sodium on the outside, This creates a relative negative charge inside the cell. So inside the cytoplasm relative to outside the cell is a little bit negative. We're going to see in physiology it's about negative 40 to negative 70 millivolts, depending on what cell we're looking at. And we call this negative charge inside the cell, we call that the resting membrane potential. You can see there in red. So I just want to introduce you to those terms so that when you get to physiology, it makes sense that you've heard about the difference between potassium and sodium concentrations inside and outside the cell, and how that leads to a difference in charge. The organelles, of course, are what are like little organs inside the cell. And I think I said previously that the microvilli were organelles. That's not correct. The microvilli are just extensions of the plasma membrane. So we're going to get into talking about the organelles here. And you have two basic types, as I mentioned before, those that have a membrane and those that do not have a membrane. Those that have a membrane, the membrane is really important for isolating the process. So let's say, for example, you're talking about the mitochondria, and the mitochondria are trying to produce ATP. Well, they need to be able to hold on to all of the molecules that are being formed and used to make energy. If those molecules were to escape into the cytosol, then they wouldn't be able to produce the energy that they're making. And other organelles are the same way. You want to be able to separate what is being made in these organelles from the rest of the cell. Maybe it's something that's dangerous to the cell. Maybe it's something that the cell would use up, and you don't want the cell to use it up because you want it to be transported to other cells. So you have to have a membrane around those organs in order to separate it from the rest of the cell. There are some organelles that do not have a membrane, and we'll talk about those as well. So let's talk first about the non-membranous organelles. And I already mentioned this a little bit with the cytoskeleton and the microvilli. Microvilli were just one example of how the cytoskeleton can be used inside the cell. But in general, we talk about four primary types of filaments that make up the cytoskeleton. So remember, I was saying earlier that the cytoskeleton is kind of like, think of it as the skeleton inside the cell, in the sense that it gives support and shape to the cell. But it does other things as well that we'll see here in a minute. So let's look at four types of filaments. Now, filaments are really just, think of them as strands of protein that have relatively different amounts of thickness, different amounts of strength. So some of them are more rigid, some of them are more flexible. And so they have different purposes depending on how thick and how flexible they are. So microfilaments, as you might imagine from the term micro, these are the smallest filaments. Often we call them thin filaments, and you're going to see this especially when we get to muscle, and we talk about how do muscles contract, and we will talk about thin filaments, and we'll talk about thick filaments, which you see down here as number 3. And the thin filaments slide against the thick filaments to cause contraction of muscle. So we'll talk about that once we get to the muscle part of the course. For now, just recognize that thin filaments are one of these four primary types of filaments. They're composed of actin. That's going to be important when we get to muscle too. So microfilaments, also called thin filaments, are composed of actin, and they work with myosin to help cells contract. Intermediate filaments would be the ones that are kind of medium in size, and these ones are kind of like the railway. They're kind of like the transport system. So although they do provide some support for the cell, they become a system of transport so that you can actually transport proteins and hormones and other things that are made inside the cell from one side of the cell to the other side of the cell by using these filaments as a kind of like a railway system. Thick filaments I already mentioned. These are composed of myosin. So you're going to hear when we get to the muscle chapter of actin filaments moving on myosin filaments. These are the thick and the thin filaments. So they're involved with muscle contraction. Microtubules are a little bit bigger, and we'll see some of the structures that they produce inside the cell. They are used to create the shape of the cell, as well as to give some specialized movement that we'll see here in a minute. They help with the movement of chromosomes during mitosis, and they're going to form specialized structures like cilia and flagella, which we'll talk about. So 4 primary types depending on their size, and they have different functions related to that size. Excuse me. So let's talk briefly about the larger size, the microtubule structures. And I mentioned that they could form cilia or flagella. You might remember flagella from high school biology or even earlier, that there are some one-celled organisms that have a flagella that help them propel, through pond water or whatever. In the human body, there's really only one cell that has a flagella, and that's the sperm cell. You can see the picture over here. You have kind of the head of the sperm, and then you have the tail, and that allows the sperm to swim through the uterus to the fallopian tube and to meet up with the egg. Cilia are sometimes confused with microvilli because they are kind of like finger or hair-like projections that come off of the cell. But microvilli, remember, are their purpose is for absorption, to increase surface area for more absorption. And they're going to be smaller, shorter. They're not going to project out quite as far. Cilia are actually produced by microtubules, so the filaments that are used are different. And they're going to be longer. They really are not built to help with reabsorption. They're built to help with movement. So, for example, in the human body, we have cilia in our respiratory tract because we produce mucus in our respiratory tract. And we have to have a way of getting that mucus up and out of the respiratory tract. And the cilia on respiratory epithelial cells, they help move that mucus up and out of the lungs. And so that's one of the primary places we see it. In the female reproductive tract, we also see cilia in the fallopian tubes, and that helps to move the egg that has been released from the ovary through the fallopian tube and towards the uterus so that it can unite with a sperm. So those are really the 2 places in the human body where we find cilia. Just remember the difference between microvilli and... Another microtubular structure is something called the centriole. You might remember this from high school biology as well, where they are they kind of function almost as more like that railway system where they direct the movement of chromosomes from one cell into another cell. So as the cell divides and splits into 2, you have to split up the genetic material. You have to give half the chromosomes to one cell and half to the other. And so that the movement of the chromosomes from one cell to the new cell is directed by these centrioles. Excuse me. Another non-membranous, meaning it doesn't have a membrane organelle, is called the ribosome. And ribosomes produce proteins. That's their primary purpose. Ribosomes have 2 subunits. You can see here in the picture, there's a large unit and a small subunit. And the translation of DNA occurs where those 2 units meet, and proteins are made from the DNA code. You can have ribosomes that are free in the cytosol, or you can have ribosomes that are attached to endoplasmic reticulum. And you can see here in the picture these kind of strands that you see here. This is the endoplasmic reticulum, and you can see the little black dots sitting on top of it. Those are ribosomes on top of the endoplasmic reticulum, so we would call that rough endoplasmic reticulum. But you can also see that there are ribosomes kind of free floating out here in the cytosol as well, making proteins. So next we'll talk about the membranous organelles. We'll start with mitochondria, which most people are familiar with as kind of the powerhouse of the cell. And we call it that because it literally produces power, energy. that our body needs for all the things that it does. We need ATP, for example, for muscle contraction. We need ATP to run our ion pumps to pump sodium out of cells and potassium into cells. You're going to see ATP coming up over and over in physiology as the primary energy source for pretty much everything that the body does. So, and a lot of that ATP is going to be produced in the mitochondria. At least the mitochondria gives you the most bang for your buck as far as producing ATP. So you can see here from the picture that we've got actually two membranes. You've got this kind of outer membrane here, and then you've got another inner membrane on the inside. And the folds that you see, if you're looking at these folds in here, those are called the cristae. And this becomes important for moving electrons across the inner membrane, and it's the movement of those electrons from one molecule to another that releases energy, and that energy is eventually captured as ATP. And again, we'll talk more about that in physiology. For now, just realize that in order for the mitochondria to produce sufficient amounts of ATP, you have to use oxygen. In fact, we're going to find out in physiology, this is the reason you breathe. You breathe so that you can use oxygen in the mitochondria to make ATP. Oxygen becomes the final electron acceptor. So after that electron has bounced from one molecule to another molecule, and every time it bounces, it releases a little bit of energy, and that energy is captured as ATP. But eventually there has to be some molecule that captures that electron and keeps it and holds it. Free electrons floating around in our body are not good for our tissues. They can actually damage our tissues. We call them free radicals. So we have to have some molecule that will accept those electrons at the end of the process. And oxygen is the final electron acceptor. So if we don't have oxygen, we really can't make very much ATP, because this process that's going on inside the mitochondria will not happen without oxygen. So we use oxygen to make ATP, but we produce carbon dioxide in the process. So this is the reason why when you breathe in oxygen, and when you breathe out carbon dioxide. We want to take oxygen into the body to be the final electron acceptor. Then we're producing excess CO2 that we have to get rid of. So when we exhale, we get rid of the excess CO2 that we produce. What's interesting about the mitochondria is they control their own reproduction, meaning they decide when they make more of themselves depending on the energy requirements of the cell. So as the energy requirements of the cell increase, like let's say a muscle cell that's repeatedly exercised, let's say you've got a bodybuilder or an athlete who's working on building muscle, as they continue to build that muscle, the mitochondria will realize they need more energy, and they will reproduce themselves, and you will find many more mitochondria 6 months after you started working out than you had before. The nucleus is typically what we call the center of the cell. It's not always actually physically in the center, but it is the part of the cell that contains the genetic material. So the DNA in forms of chromatin and chromosomes, which we'll see here in a minute, those are housed inside the nucleus. The nucleus of most cells is also surrounded by its own membrane, and that membrane is important because it allows only certain things to come in and out of the nucleus. You want to be very selective about what can come in and change the DNA or activate DNA or deactivate DNA, but you also need a way for proteins that are made inside the nucleus to get out into the cytoplasm so that it can be packaged and sent to other cells in the body. So the nucleus is important for all of that. Most cells will have one nucleus. We're going to have a couple of examples when that's not the case. Skeletal muscle or voluntary striated muscle that we'll see at the end of the next chapter briefly has many nuclei per cell, so we call that multinucleated. Red blood cells, on the other hand, actually have no nucleus, and that's because they don't produce any proteins, and we'll talk about that when we get to the blood chapter. So skeletal muscle is multinucleated, and red blood cells have no nuclei. Otherwise, for the most part, we'll see there's a few other multinucleated cells, but for the most part, we'll see most cells have one nucleus. So I already mentioned the nuclear envelope, or the kind of membrane that surrounds the nucleus. Remember, we're talking about membranous organelles now. And I mentioned that you had to have a way of material coming into and out of the nucleus, and that's going to happen through these nuclear pores. They're going to control movement of material in and out of the nucleus. So we know that the nucleus contains the DNA, which is kind of the blueprint for everything that's produced in your body. But that DNA can be in different forms. So I think most of the time when we think of DNA, we think of chromosomes. We all learned we have 46 chromosomes, or 23 pairs of chromosomes. And that's true. And often when the cell is getting ready to divide, chromosomes do form. But in the life of a cell, what you would see if you looked inside the cell, sometimes you might see chromosomes, which would be kind of tightly packed genetic material that's tightly coiled so you could actually see them. And you've probably seen pictures. They kind of look like an X because they're two pairs of the same chromosome. Or not two pairs, they're a pair of the same chromosome. And that's what we think of when we think of chromosome, and that's because it's tightly bound to a protein called histone. So that means when we stain the cell, we can actually see those chromosomes, and I'm sure you've seen pictures of chromosomes inside a cell. But sometimes the genetic material is not that tightly wound. It's more loosely bound to the histones, and it doesn't change the genetic material, but it would change the appearance if you looked on the microscope after staining. And so when it's loosely bound instead of tightly bound, instead of calling it chromosomes, we call it chromatin. And then nucleoli are kind of dark spots within the nucleus, and that's where ribosomes are being made, so that proteins can be made inside the nucleus as well. So here's just a picture showing the difference between chromatin and chromosomes. So if you look inside this nucleus here, you wouldn't see any tightly bound, organized chromosomes like we're used to seeing. You would just kind of see dark strands of material, and that's because it's loosely bound to the that histone. You can see here that histone protein. Chromosomes, on the other hand, if you looked in the cell, they kind of look like that. And that's just because the DNA is more tightly wound, so it looks thicker under the microscope, and you can see it. Okay, endoplasmic reticulum, or ER, comes in two forms. It can be smooth or it can be rough. You see the rough on this side because the ribosomes are attached to it, and I mentioned that earlier. And you see on this side, this is the smooth because there's no ribosomes attached. So endoplasmic reticulum is basically a membrane system within the cytoplasm that allows for usually different proteins that are being made to be modified to be made from these. You see the ribosomes are sitting there, so they're going to be making proteins, and those proteins will go into the cisternae, kind of that fold system that you see there. Other things like lipids and carbohydrates may also be made, but they're going to transport that material that's being made from the ribosomes. It will go through that network, and it will move along and kind of helps it move from one part of the cell to another part of the cell. It's also involved with the detoxification of drugs and toxins. And you can see that it stores some substances as well from the cytosol. But I think probably what's the main takeaway from endoplasmic reticulum is the synthesis here of proteins, lipids, and carbohydrates, the transport of substances, and the detox of drugs. And remember the difference between rough and smooth endoplasmic reticulum. So rough ER, I mentioned, is covered with ribosomes, and smooth ER has no ribosomes. So you can imagine that the rough ER is going to be synthesizing proteins, and you can kind of see here in the picture, you've got your ribosomes here. They are reading the messenger RNA, and they are producing proteins. Now those proteins then have to get kind of cut and packaged and formed, and you see all that is happening inside the endoplasmic reticulum. And eventually that still unfinished protein is going to be released, kind of butted off from the endoplasmic reticulum, and it's going to move to the Golgi apparatus, which we'll talk about next. Now, if it's a smooth endoplasmic reticulum, that means there's no ribosomes. If there's no ribosomes, that means there's no protein synthesis. But other things are still being made in there, like lipids, steroids, carbohydrates. We're going to see in muscle that there's a specialized type of endoplasmic reticulum, of smooth endoplasmic reticulum called sarcoplasmic reticulum, and that's going to store calcium ions. So remember we were talking in the previous slide that endoplasmic reticulum can store substances from the cytoplasm. This is one example of that in muscle. The endoplasmic reticulum stores calcium ions. And then we talked about the inactivation of toxins as well. Okay, so once the not quite fully finished proteins or other substances are released from the endoplasmic reticulum, they will travel to the Golgi apparatus, where they go through another series of cisternae, and they get further matured and further developed and eventually packaged into what will be the final product. Sometimes they are packaged with enzymes that are needed to activate the proteins or hormones. But what you get in the end, after they've gone through the cisternae, you get kind of a ready for release package, typically left in a vesicle, and then that vesicle will travel to the plasma membrane. Will fuse with the plasma membrane, and you will get exocytosis of that protein, for example, that was made. Okay, lysosomes basically are cleanup organelles. They're going to receive either foreign material. They might receive bacteria, for example. If you're a macrophage and you're going through the body eating bacteria, you have to have a way of destroying that bacteria. That would happen in the lysosome. Proteins that need to be degraded and kind of recycled, that would happen in the lysosome. So they're kind of the cleanup crew. And so they contain enzymes within them that can break down proteins. So that would be able to kill bacteria, for example. That would be able to recycle damaged or unused proteins and recycle the amino acids back into the cytosol to be used to make new proteins. The body also uses it, however, for types of cell death. So there are times, for example, when the whole cell is not working properly, the whole cell has become damaged or old, and you need a way of removing that dead tissue. You can't just let damaged and dead cells build on top of each other. There's not enough space. And so sometimes the body will actually release the enzymes from the lysosome onto itself and will cause the cell to be kind of dissolved by those enzymes. And we call that apoptosis, or what some people have called cell suicide. So that also occurs through the means of the lysosome. Muscle atrophy is similar. If you have ever, for example, damaged a muscle or a joint, and you've had to have a cast put on or a brace put on for a week or 2 or 6, you'll notice that the muscle gets smaller and smaller because you're not using it. It's not that you, well, we'll just say what happens in that case when you're not using that muscle and that muscle gets smaller. Part of what's happening is the lysosomes are dissolving some of the proteins inside those muscle fibers, and so they're getting smaller and smaller. And of course, once you get function of that and you exercise that, maybe do some therapy, you'll be able to build that muscle back up. But lysosomes are involved in that process of muscle atrophy when you don't use a muscle. Peroxisomes are kind of along the same lines as lysosomes, but they're kind of specialized because they use hydrogen peroxide as their agent for breaking things down, and so they can't break down everything. They're not kind of as strong, let's say, as lysosomes. They're not as versatile as a lysosome, but they are used to break down fatty acids, and they're used to neutralize some toxins in the liver. They get their name, however, from the hydrogen peroxide that's used to break things down. There are also attachments between cells. So we've been talking about what goes on inside the cell, but most of the tissues in our body, or all of the tissues really in our body, are a combination of multiple cells that are stuck together. And they have to be held together tight enough that they can function as one unit. So what is it that holds one cell, that holds it, that attaches it to surrounding cells? And that's what we're going to talk about here just briefly for a minute. There are some molecules that we call cell adhesion molecules, or CAMs, and I think we'll see a picture here in a minute of what that looks like. But these are molecules that are kind of hanging off the cell membrane, and so they attach one cell membrane to a neighboring cell membrane, and then that way the two cells are held together. Then there's also cell junctions, which tend to hold cells together more tightly, or they serve a specialized function. So for example, gap junctions that you can see here, gap junctions are kind of like, they're attachments. They hold cells together, yes, that's true, but they kind of form a hole in between the two cells, or a tunnel in between the two cells, so that material can move from one cell directly into a neighboring cell. This becomes important for tissue that has to communicate rapidly between all the cells in the tissue. So for example, heart tissue, if you want your heart to contract, let's say you want your left ventricle to contract pretty much at the same time, then you need to get the signal to contract. You need to get that message to all the cells in that left ventricle at the same time, more or less. And the way that your body does that is through these gap junctions. They're just basically tunnels. So once calcium enters one cell, it diffuses into all the surrounding cells through the gap junctions, and then the signal to contract occurs pretty much spontaneously. Other cell junctions have other specialized functions. So for example, tight junctions, just like they sound, are junctions that hold one cell to a neighboring cell very, very tight. In fact, so tight that not even a molecule of water can pass in between the cells. Now, this becomes really important. You can imagine in places like the urinary tract or the gastrointestinal system. If you've got liquid stool passing through your intestines, you don't want that liquid stool to be able to leak in between your cells and get into your deeper tissues. You want to have basically a waterproof seal that keeps all of that liquid stool inside the lumen of the small intestine. And we do that through tight junctions, so tight that not even water can pass through in most instances. And we see that in the kidney and the gastrointestinal system. Okay, just to give you a quick picture of what I'm talking about, you can see that this first one labeled B, that's the gap junctions, and you can see that they hold this cell to this cell, so they're connected, but they connect it through almost a tunnel, so that if there was a substance here, it could pass through that tunnel and travel to the neighboring cell with basically no difficulty. And as I mentioned, that's really important for cells that need rapid communication between the two of them. So we use gap junctions there. However, sometimes we need a junction that's much tighter that won't even allow anything to pass in between, and that would be our tight junctions here. They just bring the cells so close that water molecules, for example, trying to get through would not be able to because the junction is so tight.